Bueno, pues buenas tardes a todos y a todas. Vamos a reanudar esta sesión científica y lo vamos a hacer también con unos ponentes de altura. Vamos a comenzar con John Vincent, que es doctor por la Universidad College London, Inglaterra. En 1995 se trasladó a Toronto y trabajó primero en el Departamento de Psiquiatría de la Universidad de Toronto y posteriormente en el Departamento de Genética y Biología Genómica del Hospital For Sick Children de la misma ciudad. En la actualidad es investigador científico del Centro para la Adicción y Salud Mental de Toronto. El título de su ponencia: Modificaciones posttraduccionales en la región N-terminal.
Okay.
John Vincent tiene la palabra.
Hola. Buenas tardes. So, the title of my talk is, the official title is N-terminal Post-Translational Modifications of MECP2 and their role in Rett syndrome, but I've given it a snappier title, Lost in Post-Translation. So, I think I can use this. So my main research focus for some time has been looking for genes involved in autism and other neurodevelopmental disorders, as well as psychiatric disorders, and once identifying the genes, then studying them. So my introduction to Rett syndrome and MECP2 came through my interest in autism and other neuropsychiatric disorders. And I first worked on MECP2 back in the early 2000s and was involved in the identification of the E1 isoform. So currently my main interests are looking at the differences between the E1 and E2 isoforms. And secondly, what is it that rare variants in MECP2 can tell us about its function and its role in disease? So going back a step, I talk about isoforms, but what I really mean is the MECP2 gene encodes the MECP2 protein, but there are two different flavors of MECP2, and these are determined by the way the coded regions within the genome are stitched together. So the MECP2 gene, as it's strung along the X chromosome, has 4 exons, and in the process of converting the gene into protein, the exons are stitched together in mRNA, and it turns out, as we found in 2004, that it can be stitched together in several different ways. So at the time, the canonical version just had exons 2, 3, and 4 making up the coding region of the gene. And then the second version, which we now call E1, skipped exon 2 and just has coding material from exon 1, 3, and 4. So these two flavors of the MECP2 protein are very similar. They're very similar in length, 498 amino acids long versus 486 amino acids long. The only difference is what we call the N-terminal, which is the first part of the gene to be translated into the protein, which would be on our left-hand side. So the protein would be translated that way. So there are different types of mutation in Rett syndrome. I just thought I'd contrast two of the main types first, missense and nonsense. Nonsense mutations it's the name we give where a mutation changes a codon that would encode one amino acid, but the codon would then change to a stop codon, and that would prematurely truncate the protein. And these nonsense mutations can occur anywhere along the MECP2 gene, and they're very similar. There may be differences depending how far along, how much of the protein gets truncated, or whether the mutation itself triggers a process that degrades the mRNA as well. On the other hand, missense mutations change one amino acid codon for another amino acid codon. So this, at the specific site of the mutation, would potentially change the chemical or biochemical properties of MECP2 at that specific site. And as a geneticist, nonsense mutations and other truncating mutations, we like them because they're very clear. They tell us that there's something very seriously wrong here. And that if we're trying to find a new gene, that's often very good evidence. But as a molecular biologist, missense mutations, I would say, are far more interesting and far more useful for trying to discover fundamental things about the properties of the protein we're interested in. So this is a depiction of the MECP2 protein, and as has already been mentioned, there are several well-characterized domains of the protein. MBD is the methyl CpG binding domain. TRD is the transcription repression domain. In between, there's something called just the intervening domain. So as I mentioned, nonsense mutations tend to occur anywhere, whereas missense mutations tend to cluster in either the methyl binding domain or the transcription repression domain. And it's roughly a third of Rett girls will have a missense mutation, roughly a third will have nonsense mutations. So what I'm particularly interested in the really rare mutations. And one of these is this ALA2-VAL mutation. As you can see, the frequency is just 0.8% of Rett girls. So to my knowledge, only four girls have been identified with Rett syndrome in this mutation, and one with intellectual disability and the ALA2-VAL. So this is right at the beginning of the protein and only at the beginning of the E1 isoform. So why is this interesting? It's what I think is really interesting is that it only affects the E1 isoform, not the E2. And it's long been, we've known about this E1 protein, but it wasn't clear initially that what the relationship was between E1 and E2, and we hypothesized when we found it that E1 would be more relevant to Rett syndrome than E2. And that's been borne out by mouse studies where Janine is involved in this, knocking out the E1 isoform specifically or knocking out the E2 isoform specifically. If you knock out E1, you get a Rett-like phenotype. If you knock out E2, you get a pretty normal mouse. Secondly, we found mutations in this coding region in exon 1, but as far as I know, maybe Alan can confirm, no mutations have ever been found in exon 2. So it still stands. So we're pretty confident that E1 is more relevant. But they're so similar. So what is the difference between these two proteins? There's no known functional domain at the N-terminal region, this beginning region of E1, and the same is true for E2. So why is this mutation causing Rett syndrome? So this is just what it looks like at this protein sequence level. You have this in the red box is what is in common between E1 and E2 isoforms. And the white is the E1 sequence, the blue is the E2 sequence, and here we have E1 with the ALA2-Val mutation. So the second position changes an alanine amino acid to a valine amino acid. So our hypothesis at the time was that perhaps it's affecting a process called post-translational modification. Now, this is something that most proteins undergo, and in particular at the N-terminal end. All proteins that are made in eukaryotes will have a methionine amino acid at the N-terminal end. But this is excised during the process when the protein is manufactured in the cell. Following the excision of that N-terminal methionine, typically a protein would then be acetylated, and that acetyl group at the end of the protein would protect, it would prevent a process called ubiquination, and that would prevent the protein being degraded. So one mechanism would be that the mutation is somehow disrupting this post-translational modification and allowing MECP2 to be degraded too rapidly, or potentially an effect on the post-translational modification would be causing the protein to be sent to the wrong subcellular organelle or compartment. So what is interesting is when you look at all proteins, you see that these post-translational modifications are not always the same. The efficiency is different depending on the next amino acid after the methionine. So if you have an alanine as the next amino acid, it's 100% efficient. The methionine is excised, but if you have valine, it's only 50%. And a French group came up with an algorithm that would predict what happens to different proteins, and that sort of supported our idea. Here we have the E1 wild type, the alanine here. It's telling us the methionine is removed and then acetylated, and it's predicting a half-life for the protein of 220 hours. If you change that alanine for a valine, as in these Rett girls, it's predicting methionine excision, but no acetylation, and it's predicting a much shorter half-life for the protein. E2 is, using the same prediction, ends up being very much like this mutant E1. It predicts methionine excision, no acetylation, and a shorter half-life. So now to test this hypothesis, we use protein sequencing approach called mass spectrometry. So here we have the protein sequence for E1, and then these are the alignments of the peptides that we found. The A indicates an acetyl group, and you see there are no reads that extend as far as this methionine. That's because all the methionine has been chopped off, as predicted. What was never predicted was what we're seeing here, that there's actually chew-back of this polyalanine stretch of the E1 protein. And as far as I know, that's never been reported anywhere, not just for MECP2, but for any protein. And in the case where you switch that second alanine for valine, we see there's some evidence for excision of the methionine, but it's not complete. There's some evidence of acetylation at the end as well, but that's not complete either. And similar story for E2. We see incomplete excision of methionine, but we do see acetylation. We don't see the unacetylated product. But just going back a second, we don't see this two-back that we were getting with the wild type E1. So does this affect the function of the localization of MECP2? We don't think so. This is wild type MECP2 being localized nicely to the nucleus in cells and clustering the chromatin into these nice chromocenters. Same thing with the ALA2 val mutation, so there seems to be no difference there. So we did try to look at the stability of the protein using something called the cyclohexamide chase assay. So you're injecting your protein into a cell, and then basically, or you're putting a construct into the cell that makes your protein, either the wild type or the mutant, and then you're kind of telling the cell to stop making any protein. And the protein has a fluorescent tag on it, and then you just see how long that tag glows green, basically. And you follow that and see how long the protein stays around. And what we see, this is called a Western blot, and it shows the protein, the E1 protein, at 0 hours, then 24, then 48. By 48 hours, it's almost gone. The ALA2 val mutation is there at 0 hours, but it's gone almost immediately. E2 seems to stick around possibly even longer than the E1 isoform, so perhaps it's even more stable. And this is borne out in different cell lines, different conditions, so we're pretty confident about this. So in summary, for this mutation, we think that Rett syndrome is being caused by this premature degradation of the E1 version of the protein in these girls' cells. And this premature degradation is leading to the not enough MECP2 protein being around in their cells. So I'm going to change tack a little bit, move on to a second topic. It's kind of well established in X-linked diseases that whether it's dominant or recessive, there's a high degree of variability in the phenotype, in the expressivity, and the incomplete or reduced penetrance of the disease in females. And it's now suggested that we don't use the terms recessive or dominant when we're talking about X-linked diseases, and that we just use the term X-linked. So what we see is a continuum in the penetrance between the two sexes for X-linked diseases. At one end of this continuum, you see female penetrance is very low. All the affecteds are male. In the middle of this, you see penetrance high in both, but more severe in males than females. And then the other end of this spectrum, you see penetrance high is high in both males and females, but severity is higher for males who would then die either in gestation or early infancy. So that would echo the scenario we see for a lot of Rett syndrome mutations. But boys can have mutations in MECP2 and survive. In some cases, like for threonine 158 methionine, they would have this very severe phenotype. It's not Rett, it's a neonatal encephalopathy, and death typically occurs fairly soon after birth. But other mutations in this gene seem to have very mild or no consequence in females, but there is a clear clinical phenotype in boys. And so, I mean, I'm interested in this, and I realize it's not Rett per se, but this can tell us more about the MECP2 protein. So I'm going to point out this mutation here, proline 152 arginine. It's one of the more common mutations in the methyl binding domain. It causes Rett syndrome in girls. Another mutation at the same amino acid, proline 152, but changing it to histidine, has been reported in this 40-year-old male who has intellectual disability, who has bipolar disorder. He suffered quite a significant cognitive regression in his 20s. Now he can't walk. And then a third mutation, again at the same position, but proline converting to alanine in a family, a 10-year-old daughter affected with obesity and pervasive developmental disorder similar to autism, and a learning disorder in her father who carries the same mutation, as well as obesity. So something very different about that happens when different amino acids are substituting at this position. So what we see when we model the protein on a strand of DNA here, you see the methyl binding domain has, there are some of the mutations line up very close to the DNA. They're actually involved in binding to the DNA, whereas other mutations at the other end of this methyl binding domain, and it seems we get very different effect in the cells when we put proteins with these mutations into the cells. So again, this is a wild-type nucleus. You see the chromocenters coming up nicely with threonine 158 methionine, arginine 111 glycine, these mutations that the position is right on the DNA. You see hardly any DNA binding at all, no chromocenters. But at this P152 distal position in the methyl binding domain, it's doing something else. You're getting chromocenters, but they're much smaller, and there are much more of them, or many more of them. Similarly for the P152H and P152 alanine, but the effect is less. So, and you see it in this graphical form. And going back to what Alan was talking earlier about studying symptoms in males, you can have very, very severe phenotype. This is using Alan Peirce's clinical severity score. You can have these very, very severe phenotypes, like with the threonine 158 methionine mutation, all the way to this proline 152 alanine, which is very, very mild. And these scores seem to correlate nicely with what we're seeing at the cellular level in the nucleus. So moving on from that, particularly with that P152 histidine chap who has bipolar disorder, it started making me think about a lot of other phenotypes that have been reported with MECP2 mutations. One of the most commonly reported is alanine 140-valine, and schizophrenia has been reported a lot of times in these patients. And recently, a paper came out with a mutation in serine 164 isoleucine in a pair of dizygotic twins, both with Asperger's syndrome, one who also developed childhood onset schizophrenia. So these are both in the methyl binding domain. We also see something in the intervening domain. We see there was a report a couple of years ago in a schizophrenia study of a female who has a de novo mutation. It's not in either of her parents. It occurred spontaneously, and she has schizophrenia. And then, more recently, we found this family from Pakistan who have the mutation in the same position in the intervening domain, but changing arginine not to cysteine, but to histidine. And this family segregates intellectual disability and schizophrenia. And this is a pedigree of the family. It's complicated, but in my opinion, it's very, very interesting. You see the grandfather up here is the obligate carrier of the mutation. He's reported to have, he's deceased now, he's reported to have mild intellectual disability and psychosis. He passed it on to a daughter who's was affected only with depression. She passed it on to 3 of her children, all of whom have mild intellectual disability, 2 of whom also have psychosis. And also 2 of her female siblings have intellectual disability, or both intellectual disability and schizophrenia. But also in this family, you have these 2 brothers who have schizophrenia, but they don't have the mutation. So that kind of made us want to go back to the family and actually do a little bit more detailed clinical analysis. And talking to the family, what we see with all the individuals who carry the mutation, they, when they were infants, they were developing normally. The boy around age 4 or 5, he was fine, but then he started regressing cognitively and he never went to school. The girls started similar kind of thing, but their regression started around age 9. And as you can see, 3 out of 4 of them also developed psychosis. So we're also interested in this family to see what other mutations, variants could be in this family. We're not 100% sure. There could be another mutation somewhere that is predisposing the schizophrenia as well. We just don't know at this stage. But this is where the mutation is in MECP2. The intervening domain contains this 80 hook domain that was mentioned earlier. The amino acid sequence all around here is very, very positively charged. DNA is negatively charged. So where 80 hook domains are known to bind to DNA, and we think this disruption of this positively charged arginine for either histidine or cysteine is disrupting this binding. And we did some assays using DNA probes and the wild type or mutant protein. And we see this very nice effect. You see very little unbound DNA with the wild type protein, some unbound DNA with the R190 histidine, but virtually no bound DNA with the R190 cysteine mutation. And a similar kind of assay, a more direct assay, just using fluorescence, shows a very similar thing. And this is kind of logical in terms of you would expect histidine to be more like arginine because they're both positively charged, whereas cysteine is a neutral amino acid. So it makes biochemical sense that it would be a severer phenotype, or certainly a severer cellular phenotype. And this is also true rather than just a random DNA probe that has AT-rich DNA. We use the BDNF promoter sequence as well and see the same binding defect here. So that was what we expected. What we didn't expect was that we also see a phenotype similar to what we saw with the arginine 152 mutations, this chromatin clustering defect. You see smaller but more numerous chromocenters. So it's not just this AT hook activity that's affected. There's some chromatin compaction deficits going on here as well. So I'm going to leave it at that. Just to summarize by saying that the mutations in this AT hook domain appear to affect both DNA binding and chromatin clustering. More generally, these different types of missense mutations in and around the methyl binding domain can have very different effects on chromatin binding, DNA binding, and chromatin clustering. And then more broadly, that these mutations may affect males and females differently in terms of the penetrance, the expressivity, and severity. And you may see very different clinical phenotypes. So, for instance, more psychiatric phenotypes coming up in patients with MECP2 mutations. Not Rett syndrome. What I'm trying to say is this gene is more important, is not just important for Rett syndrome. There's more going on here. So finally, I'd just like to thank you all for your patience. Thank particularly my student Timur Sheikh, who's done some fantastic work here, and my collaborators, obviously Juan Alcio, who's very much involved. And also in particular, I'd like to thank the Ontario Rett Syndrome Association for supporting our work. Thank you very much.